things over to Winfried Weissenhorn, the director of the IBS, who will formally introduce today's speaker.
Okay, thank you very much, Carlo. I'm really happy to have Andrea here today to present the development of the Moderna vaccine. Andrea has a long career in structural biology, which he started here at IBS when he did his PhD in the lab of Otto Diedeberg. Then after his PhD, Andrea moved to Don Wiley's lab at Harvard University, where he worked on glycoproteins from herpes virus and other viruses. And then after his PhD, he moved back to Italy to Merck, where he was a project leader and then head of structural biology until I think 2010. And in 2010, he moved back to Cambridge, and where he became global head of structural biology of Novartis. And if I remember it correctly, this section of Novartis was then bought by GSK, and you stayed 1 year at GSK in Boston, in Cambridge, and then joined in 2017 Moderna, where everything began, the new era of vaccine development. And we're looking very much forward to your presentation. Thank you for taking the time.
Thank you, merci. Thank you, Winfried. Deux mots en français, ça me fait beaucoup plaisir de présenter pour l'IBS où j'ai fait ma thèse de doctorat. Merci beaucoup, Winfried, pour l'opportunité de présenter les données. Je vais parler en anglais. Non, en français.
Yes, please.
Give me just a second to put it on presentation mode. Okay, hopefully you can see my screen. So what I'm going to do is I'm going to present how we went through the development of the Moderna COVID-19 vaccine. But initially, I will give some background on the mRNA technology, how it's can be applied to vaccine development, the efforts, again, preclinical and clinical on the COVID-19 vaccine, and then some of the work we are doing to tackle the new variants that are now circulating. So just as a disclaimer, the COVID-19 vaccine, the Moderna COVID-19 vaccine is authorized under emergency use authorization, so it's not an approved vaccine, but is authorized to be used in under emergency situation for a population from 18 years to older. And so that authorization can be taken away anytime. So the technology is based on the delivery of messenger RNA encapsulated in lipid nanoparticles into the cytoplasm of cells. Once in the cells and released from the endosomes, the mRNA engages the ribosomes and gets translated into proteins. As you can imagine, you can express different proteins, either soluble, membrane-bound, or also localized in different compartments. Therefore, the technology offers many opportunities in terms of therapeutic area that you can tackle. But when you express a protein that is a viral protein, bacterial, or a parasite, so from a pathogen, this is recognized as a foreign protein, resulting in an immune response. Therefore, the technology is amenable to vaccine development. The two key features are the messenger RNA and the lipid nanoparticle. The mRNA is a standard mRNA, as we all have in our cells. The two untranslated regions of the 5' UTR and the 3' UTR. There is a long polyalanine tail, and between the two UTRs, there is the open reading frame that encodes for the protein of interest. The mRNA is capped at the 5' end. All the elements are optimized to have optimal expression and also stability of the mRNA. One modification that we have introduced is a replacement of a uridine with a pseudouridine. And this is to prevent excessive engagement of toll-like receptors, innate immunity that may result in the shutdown of protein expression. The second element is the lipid nanoparticle that encapsulates the messenger RNA and protects it. And it's critical, of course, for the delivery of the mRNA inside the cells. There is different components that play a key role, PEG lipid, the phospholipid, sterol, and aminolipids. And they're relevant for the stability of the lipid nanoparticle, but also for the release of the mRNA once the LNP is within the cells and within the endosomes. And depending on the process, you can clearly change the properties of the LNPs, and also by changing the composition, you can target different cells. And so we have different LNPs for different applications. The LNP that we use for vaccines, for instance, is different from the LNP that we use for therapeutics. We define it as a platform technology. You start from the DNA sequences that encodes the protein of interest, then through a chemical reaction, basically you get to the mRNA. You just need the polymerase, the enzyme, buffer, the nucleotides, and these are results in your mRNA. Then you co-formulate with lipids. These are results in the mRNA lipid nanoparticle complex, and this is basically the drug product, the vaccine, that can be administered through an intramuscular injection. So you can see that all these steps are the same independently from the sequence of the protein that you want to express. The only thing that changes is the sequence of the messenger RNA, but all the other reagents and processes are conserved. So these allow for a fast product development, and clearly the technology is very well suited for a pandemic response. So once the vaccine is delivered through an intramuscular injection, then the LNP can migrate through the lymphatic to the draining lymph nodes, or dendritic cells can actually also capture the mRNA LNPs at the site of injection and bring it to the draining lymph nodes. In the antigen-presenting cells, there is expression of the protein of interest that can be then presented on the cell surface to B cells, but also on MHC class 1 and class 2, so engaging also T-cell responses. So the technology has therefore several advantages. First of all, it's highly precise. The mRNA, of course, encodes, provides the instructions that are translated in the protein of interest, and the protein has a native-like structure because, first of all, we can express, for instance, membrane-bound proteins, and also the glycosylation is closer to human glycosylation compared to recombinant technologies. Also, the technology and the mRNA is not infectious, cannot go from one cell to another, and also cannot duplicate to generate infective virus. The mRNA is rapidly degraded after delivered into the cells, and so the expression is only transient. And finally, is also non-mutational, meaning that the mRNA cannot integrate in the human genome. So as I mentioned, these are the fact that we don't need to change anything other than the sequence of the mRNA allows for really a rapid response. And that's what we did in the context of the SARS-CoV-2 pandemic. So early on, so Moderna started actually working on the mRNA technology 10 years ago. But even after the initial success or initial clinical data with the pandemic flu vaccine, invested on manufacturing. And we have our own manufacturing site outside Boston in Norwood, where we basically generate all our vaccines and all our therapeutics, mRNA LNP therapeutics for clinical studies, and now also for the mRNA COVID-19 vaccine. And if you were to get inside, you would see something that is different from regular manufacturing sites. We don't use really large bioreactors, so really the scale of the commercial manufacturer is much smaller. That allows us to have a smaller footprint, lower capital investment, and we don't really need to have a dedicated manufacturing plant for vaccine production. So several vaccines can be manufactured at the same place. So this is really advantages of the technology. And also we don't use cells. So this is again, from a bioburden perspective, it's also simpler and also safer. So we, before even start working on the COVID-19 vaccine, we had really moved several candidates to the clinic. And I will focus on the vaccine side, but we also have, of course, investment in other therapeutic areas, such as oncology, autoimmune disease, and rare disease. For the vaccines, we started with the flu pandemic. Then we moved to other respiratory viruses such as RSV and HMPV/PIV. We have also vaccines against herpes viruses. Our vaccine against cytomegalovirus that causes congenital CMV, a devastating disease, is now in late phase 2 stage, and it should start phase 3 this year. And we should get in the clinic with an EBV vaccine early next year. We also have a Zika vaccine, so we can also make complex antigens like viral-like particle, and that should move this year to phase 2 studies. I want to just mention that we also have shown that we can express in vivo monoclonal antibodies, and we have generated data in a phase 1 study for a chikungunya antibody. So even before we started working on the SARS-CoV-2 vaccine, more than 4,000 subjects had received a Moderna vaccine, and we had shown that the technology was safe and able to raise immunoresponses in humans. So in January of 2020, we heard about the cases of pneumonia in China. And of course, it was clear that there was a virus, at the beginning, not really which one, but as soon as it became clear that was a coronavirus, we decided to work on it. We had a collaboration already ongoing with the VRC and NIH on a different vaccine against the MERS coronavirus. So we knew what antigen and to basically generate, and we already had in vivo data against the MERS vaccine. So we were comfortable that we could generate a vaccine against this novel coronavirus. So as soon as we obtained the sequences of the virus, we generated the mRNA. The sequence, of course, is optimized, as I mentioned, for improved stability, expression, and translation. And the vaccine encodes for the spike protein that is stabilized in the prefusion conformation by 2 proline mutations. And what we were able to do is, from basically the sequence to really have the vaccine in vials that had passed sterility testing and so ready for a clinical study, it took only 42 days. And then before you can go in a clinical study, of course, you need all the approvals, the safe to proceed. So by March 16, so 63 days from sequence availability, we were in a phase 1 study sponsored by the NIH. So this was a record speed for vaccine development, at least in terms of getting into a phase 1 study. So I want to just point to some of the key dates in the vaccine development. Again, started in January, 63 days for first in human. We got funding, roughly $500 million from BARDA to continue development of the vaccine. We got positive phase 1 data by mid of May, and by the end of the month, we started already a phase 2 study. And end of July, so 2 months later, we started a large phase 3 efficacy study on 30,000 subjects that really was completed by the end of October with interim efficacy data by the end of November. And then, within 11 months or so from the sequence, we were able to obtain emergency use authorization from the FDA. A couple of things that I would like to point out: we didn't skip any of the clinical studies that are needed to develop a vaccine. We did phase 1, phase 2, phase 3. What really was accelerated was the financial risk allowed us to start manufacturing, and also new clinical studies at risk without having necessarily immunogenicity or efficacy data, I should say. So there were two paths that were going in parallel. There was a clinical, of course, phase 1, phase 2, phase 3 studies, but at the same time, we had to perform non-clinical preclinical experiments to demonstrate that the vaccine was immunogenic, could protect, and was also safe. And these data were critical to move to the next steps of the clinical studies. So I will summarize the key preclinical data. We initially showed that the vaccine was protective in mice, and that prompted us to test the vaccine then in non-human primate. And this is the data that I'm going to show for a number of reasons. So first of all, in NHPs, you can use doses that are more similar to what you use in humans. We tested 10 and 100 micrograms. Also, the immune system of monkeys and non-human primates are more similar, both in terms of adaptive and innate immune responses to humans. And it also was known that NHPs can be infected with the SARS-CoV-2 virus, and they've developed a mild pneumonia. So the animals received 2 doses of the vaccine 4 weeks apart. Then they were challenged 4 weeks post second immunization. And then we looked also at lung pathology because for other, some vaccines developed in the late 60s using inactivated virus, such as a vaccine against RSV. There have been cases of enhanced disease where people exposed to the virus after vaccination had worsening of the symptoms. And this had been described also in animal models for the SARS-CoV-1. So it was important to demonstrate that this was not a problem for our technology and our vaccine in NHPs before we could move to clinical studies. So we first showed that the vaccine is immunogenic, both at 100 and 100 micrograms, with a dose response, 100 giving higher titers of antibodies, and also the second dose boosting antibody titers. When we looked at the functionality, we saw that basically the antibodies were able to prevent, both at the 100 microgram and 10 microgram dose, the interaction between the receptor binding domain of the spike protein and the receptor, the ACE2 receptor. And these responses were higher than what's seen in convalescent subjects, so subjects that have recovered from a SARS-CoV-2 infection, giving you some hope that the vaccine could be protective. In terms of live virus neutralization, we also saw that the responses elicited by the vaccine were really blocking, so we're neutralizing, and higher than, again, what's seen in convalescent subjects. But sorry, the really demonstration of the efficacy, at least in this animal model, was by looking at the replicating virus post-challenge in the nose and in the lung. And what we could see that basically both 100 microgram and 10 microgram, the vaccine was fully protective in the lung. In the 10 microgram dose at day 4, there was only one animal showing some signs of replicating virus, and none at the 100 microgram. And also in the nose, first of all, we saw a decrease in compared to the placebo. By day 4, only one animal in the 10 microgram group had some replicating virus, and 100 microgram by day 2, there was none, and a blimp at day 4. So the vaccine provides an immunity that can control the virus very quickly and is fully protective in the lung and partially protective in the nose. And we also looked at the immunoresponses post-challenge, both again in the lung and the nose, and we could see that even post-challenge, there's no increase in antibody titers. So of course, it's not expressed for N, which is not part of the vaccine, but even for the spike protein, there's no increase in antibody titers. So, so there's no amnestic response, suggesting again that, you know, the virus, the challenge virus, is immediately neutralized by the antibodies. And when we looked at the pathology in the lung, we really just saw a mild inflammation on the 10 microgram, but there was no sign of either viral protein or viral RNA in the lung at either doses. So the vaccine is really providing a robust protection, and there is no sign of enhanced disease, as I mentioned. As a model for severe disease, we also performed experiments in the hamster model. And I just want to mention that, of course, we saw, again, we tested different doses and also a single dose vaccine. We were able to raise neutralizing responses for all the regimen tested. But the most important is that the vaccine was protective from weight loss, so a sign of protection from disease, and also similarly to the NHPs, we did not see, you know, virus in the lung or in the nose. So the vaccine, the data suggests that the vaccine could protect also from severe disease. So I will move now to the clinical data. So of course, the preclinical data allowed us to move to the clinical studies. We first did a phase 1 study to really have a sense of the immunogenicity and the dose of the vaccine that in different population age strata. Then we moved to a phase 2 to confirm the dose and to get, you know, more safety data. And then as I mentioned, end of July, we started a large phase 3 efficacy study that I will describe in a few minutes. There are additional studies that are ongoing. Of course, we have a pediatric study where we are testing the vaccine going from 6 months to 18 years old subjects. And that is ongoing with the data from 12 to 18. It should come sooner, hopefully bringing to an approval before the start of the fall next year, before the start of schools. And then for the 6 months to 12 years of age for end of 2021. So the phase 1 study was tested at 3 doses, going from 25 micrograms to 250 micrograms in 3 age strata, 1855, 56, 70, and above 71 years of age, 2 immunization 1 month apart. And then we looked at the immunoresponses in terms of antibody titers, neutralizing antibodies, T cells, but also, of course, the safety and tolerability of the vaccine. And the 250 micrograms after the second dose was not well tolerated. So in the older adults, we tested only 25 and 100 micrograms. So I will just show in terms of immunogenicity, the neutralization data, showing that basically at all 3 doses, we get neutralizing responses, and everyone had the neutralizing titers post the second injection, let's say day 45, for instance, with the neutralizing responses being higher on 100 microgram compared to 25, but 100 to 150 being very similar. And also, it's worth noticing that the neutralizing responses are really above the median observed for the convalescent subjects. So again, very encouraging data. And because of the tolerability and the lack of really strong differences between 100 and 250 microgram, the 100 microgram dose was selected for subsequent studies, for the phase 3 study. So this was the 18 to 55 years old subjects, but also, of course, it was important to compare across age strata. And what we saw is that basically the responses are, neutralizing responses are very similar, going from 1855, 56, 70, and above 71 years of age. So suggesting that the vaccine could confer protection for all age strata. And this is important because the elderly, of course, are the ones that are mostly affected or have more cases of severe disease. And so it was important to show that we could get robust neutralizing responses in these subjects. We also looked at the T-cell responses. This is important both for the protection, but also for enhanced disease, as Th2 responses have been associated with the enhanced disease in the RSV formalin inactivated vaccine trial. So here what we could see is that actually at all ages, we just see Th1 responses. The Th2 are not represented here, but there were none. And so the vaccine not only elicit strong T-cell responses or the Th1 phenotype, but also it should be therefore safe to be used in clinical studies and in the phase 3 study. So I'll just now move to the phase 3 study. Was an efficacy study, is observed blinded, randomized, placebo-controlled study. We have 15,000 subjects roughly in the placebo arm and 15,000 subjects in the vaccine group. The study, of course, looked at the immunogenicity and but mostly protection, and I'll describe the protection data. Of course, we also collected safety data. But one important aspect was that the trial included a population that was diverse and representative of the United States. And for that reason, actually, during development, we slowed down recruitment to make sure that actually even minorities were well represented in the study population. And that's what we were really able to do. So in the final, after at the end of recruitment, we had 60% roughly of white population, and then roughly 40% minority, with 20% of Hispanic and 10% of Black American. This distribution is very similar actually to the US population. There was also good distribution across age, and roughly 50-50 among men and women. Another important criteria that we fixed actually from the beginning in the protocol was also the, people at high risk or risk for severe disease. All the subjects in the study were at higher risk of SARS-CoV-2 infection, but we also made sure that there was really 40% that were at risk of a severe disease, either because had some underlying pathologies, or because they were above 65 years of age. Among the people at risk and with comorbidities, there was a good representation of people with diabetes, lung disease, and also cardiac disease and obesity. So I think from a perspective of recruitment, the study was actually very representative of the US population. So, the study, of course, the main and the primary endpoint was prevention of symptomatic COVID-19 disease. There were additional, secondary endpoints which were part of the study, such as prevention of severe disease and also of infection, and several others. But we were very pleased to see that at the primary efficacy analysis, when 196 cases were accumulated, that we had 185 in the placebo arm and only 11 in the group that had received 2 doses of the vaccine. So this is the primary efficacy analysis. And what we see is basically an efficacy of 94% with a 95% confidence interval. So the vaccine seems to be, highly efficacious in the prevention of COVID-19 disease. Really the efficacy was really consistent even when the population was, or the participants were stratified based on when we have a subgroup stratification, reflecting really the difference in demographics or in also underlying conditions. So, high efficacy for, all the groups. We, of course, one of the most important parameters is the prevention of severe disease. And again, at the primary efficacy analysis, we had 30 cases that were in the placebo group and none in the vaccinated group, really showing that the vaccine has a very strong protection and really may provide an opportunity to really change the direction of the pandemic. We also started getting data on the infection, which I'll describe in a second. But if you look really at the Kaplan-Meier curves, what you can see is that the accumulation really of cases starts in the placebo group even before, of course, the second dose. But in the placebo, so no, we don't have cases. So basically, there is the curve starts separating already post the first dose, so suggesting that the vaccine is providing some protection even after one dose, though the study was not designed to assess protection after one dose. These are data that we will also get from real life use of the vaccine. Also, when we looked at really at the PCR nasal swabs in subjects that had received the vaccine before they received the second dose, you can see that there are fewer cases in the vaccinated group compared to the placebo. Again, showing that the vaccine is providing roughly 70% protection even after the first dose. If you actually we generated more data, and after follow-up of 74 days, you can see that again there is a fewer cases in a smaller group that received only one dose. There are fewer cases in the vaccinated group compared to the placebo. And actually, if you start counting cases just 14 days after dose one, when you start having immunoresponses, really there is a higher efficacy. So the vaccine basically really has a strong protection. So now I'll move to the present or the future. Right, is also while the vaccination is ongoing, and it's really critical that actually keeps moving on, and that we vaccinate everyone. We are also trying to address potential concerns and risks with the new variants that are spreading across the world. And the variants that we are discussing today, of course, those are the major concern, but it's not the first time that we have a variant that is circulating. Actually, the virus that is the predominant virus until recently, the D614G, is the virus that really started circulating and increasing during 2020 to become the predominant virus circulating. Maybe we did not hear necessarily too much about it because this new variant was neutralized very well also from immunoresponses raised against the initial ancestral virus, the Wuhan virus. So what are the variants that are of concern? I mean, now there is, of course, a classification and different phylogenetic trees, but some of these variants, of course, are known for where they originated. So the B117 is the variant that started from the UK, 351 in the Republic of South Africa, P1 Brazil, and then the 429 that is the variant that started in California. So you start seeing a number of mutations that accumulate in the spike the viral S protein. Some of the mutations are in the RBD domain and some on the N-terminal domain. It's important to know that, of course, the receptor binding domain is involved in receptor binding. Also, the NTD is also important for binding to neutralizing antibodies. So mutations in this region may affect the not only neutralization by monoclonal antibodies, but also potentially by vaccines. So it was important to test this variant and see how immunoresponses raised by the current vaccines are able to neutralize this new variants. You can see that some of the, in all cases, some of the mutations are in the really the region of interaction with the ACE2 receptor. In the context of the variant that started in UK, there is only the N501Y that confers higher affinity to the ACE2, but then we have other mutations that have popped up in the other variants. The three in the receptor binding domain are the same in the South African Brazilian variant, but there is a different mutation in the variant that has started in California that is now spreading in the US. So there are also mutations in the N-terminal domain. I just want to point out that some of these variations for instance, in the N-terminal domain cluster in regions that are involved in binding to neutralizing antibodies. Therefore, having an impact on some of the antibodies that have been developed or under development. So we tested sera from 8 subjects that had received the 100 microgram dose in phase 1. And we assessed the neutralization of this sera of the different variant viruses tested as pseudoviruses. And so what we can see is that the vaccine is not, or the responses elicited by the vaccine are not really impacted by the variant that has started in the UK. But when we looked at the other variants, there is an impact on neutralization that goes from 6-fold in the context of the variant from the South Africa to 3.5 and for the Brazilian variant, and roughly between 2 and 3 for the variant that has started in California. So these variant viruses are less susceptible to neutralization, but it's important to still mention that the neutralizing responses are still high and above levels that we have seen are protective in non-human primate models. So despite there is an impact in the neutralization potency, we think that the vaccine is still protective. What may change is perhaps the durability of protection. Also, you start basically from a lower, peak neutralization. So while we have performed these experiments in vitro, but there is of course work that is ongoing in the preclinical space and clinical to really generate a vaccine that can tackle the current variants with a focus on the 351, but also future variants. And so we are advancing a vaccine that encodes for the South African spike protein in a phase 1 study as a third dose, basically as a booster of the 50 microgram dose. We're also testing a vaccine that is basically a combination of the current one and the 351 variant, again tested as a booster. And also we are testing, of course, the current vaccine as a third dose. As a third dose might still raise antibody responses and neutralizing responses and tackle the variants. And finally, we are testing combination and the single variant in as a primary series for people that have not received yet vaccine and that are seronegative. So we should see data in the next coming weeks. A couple of words on the CMC side on the products. So the vaccine can be stored at -20 degrees for 6 months and maybe longer. We also shown that now we can store the vaccine at 4 degrees between 2 and 8, so the refrigerator temperature up to 30 days, and then also 12 hours for after the vial, which is a multi-dose vial, has been thawed. So these conditions, the storage conditions, allow for distribution, across different countries and in the United States. And at this point, I think at least 70 million doses of the Moderna vaccine, which is a huge number if you compare that we started from 3,000, let's say, vaccinated subjects in the 2019. So more than 70 million doses have been administered in the US, so it's a huge number. And in terms of manufacturing, we are manufacturing both in the US. We have an alliance with Lonza, that is a large-scale manufacturer company, and also with Catalent for fill finish. And for the international market, we are working again with Lonza in Switzerland, and also we have a fill finish agreement with Rovi and now other companies as well. So the vaccine that is used in Europe is manufactured actually in Switzerland. So I'll finish here. So just to summarize, we have shown that the Moderna COVID-19 vaccine is protective in different animal models, in mice and non-human primates with no signs of ERD, and also against COVID-19 disease in hamsters. The phase 3 study has shown that the vaccine is highly efficacious in the prevention of disease and may also prevent infection. And recent data, actually even disclosed yesterday by CDC, showed that actually the vaccine is highly efficacious also in prevention of infection. More than 70 million doses have been administered in the US at this point. And we have a pediatric study ongoing to assess safety and tolerability of the vaccine in the 6 months to 70 years old subjects. And of course, there's a work ongoing to develop, upgraded version of the mRNA 1273 vaccine to really, address any potential risk with the new variants that might be circulating. So I will finish with also the acknowledgement. I mean, this of course has been a huge effort, both within Moderna, but also with lots of different partners. Of course, we had funding from CEPI at the beginning, but then, BARDA and NIH, and really critical contributions and collaborations with the NIH. I want to mention for the preclinical studies, the group of Barney Graham and also Bob Cedar for non-human primate studies, funding of BARDA, as I mentioned, but also like to acknowledge, the collaboration with the Operation Warp Speed, and also all the investigators and subjects that participated to the phase 3 safety study. So a big thank you to all of them. So I will stop with that, and I'm happy to take any questions.
Thank you very much, Andrea, for this very clear, nice presentation. I think we can switch to the questions. Laurent, you're going to present the questions.
So thank you, Andrea. So there's quite a few questions. So yeah, currently we have 21, and probably some more will come as we speak. So a bit random in terms of topics. So the easiest way is just to tell them one how they came. So if you can answer them rather briefly. Every time.
Oh, you want me to take the question? Okay.
No, I can read them out. Can you see them yourself, or?
Yeah, I can see them.
You can see them, but maybe.
Yeah. So I'll start with the first come first served. If you have,
if you read it loud, and then I.
Yeah. So could you please explain the rationale for choice of 100 microgram per dose compared with the 30 microgram per dose from Pfizer-BioNTech and the 12 microgram per dose from CureVac vaccine? Yeah. So first of all, there are two things. Of course, these were independent developments, and there are also potential difference in the technology. So the decision on the dose was made based on the immune responses that we saw in the phase 1 study and tolerability. And in our case, we didn't want to really take risk that to have a suboptimal protection. So we went for the basically highest tolerated dose and with the highest immunogenicity for the phase 3 study. When we looked at the phase 2 data where we tested 50 microgram, 100 micrograms, actually the responses are very comparable. So, in the future, if with more data and comparability data, it might be possible to go to a lower dose. But of course, for now, it's approved for 100 microgram dose. Then you have to, when it comes to other mRNA approaches, you have to look also what, where if the limitation was also in the tolerability and in the immune responses. So how did we stabilize the S protein? Yeah, so this was work that had been done previously by the NIH on other coronavirus spike proteins, and they'd shown that at least in the context of the subunit vaccine, if you wanted to have a stable molecule that raises also higher neutralizing responses, you need to stabilize it in the perfusion conformation, and they achieved that by introducing 2 proline. We had shown that the same approach was beneficial in the context of a MERS vaccine using mRNA. And so we decided that in the, when we developed the SARS-CoV-2 vaccine to go directly with the stabilized version of the vaccine. I think it's stabilization in the perfusion conformation is certainly not detrimental, and I would say is beneficial. Not all the vaccines have a stabilized spike protein at this point, but most of them have incorporated those mutations. In other viruses has been shown to be, of course, essential. Yeah, could you recapitulate how the lipid nanoparticles was devised? What specific steps, bottlenecks did it take? Will it be adapted for each of the mRNA vaccines? Yeah, so I mean, of course, that was, so the work on the LNP, on the lipid nanoparticle, has been an effort that, started 10 years ago, and through selection of the right lipid processes. And we had all kinds of challenges. When I joined, there was particle growth, you couldn't store it at 4 degrees or even -20. So there was a lot of technology development that had to be, applied to the development of the vaccine, and even the identification of a lipid that was well tolerated. So for all our vaccines, for infectious disease, we are using now the same LNP and the same composition, and that allows us, of course, to apply to not only many different vaccines, but also, as I was mentioning, for pandemic response. Of course, each vaccine company has its own now LNP with different lipid and composition. What is the maximum size? Okay, what is the maximum size of the ORF you can incorporate? There's not limitation on the size of the open reading frame that I'm aware of that can be incorporated in the lipid nanoparticle. Other companies are using a self-amplifying mRNA approach where you really have a larger cassette that is included, and so and they can do it as a lipid nanoparticle. So most of our vaccines are, so far it's on the 4,000, 5,000, as a limit base pair. So we haven't really needed to go much larger. And the other thing that maybe is worth mentioning is that we can incorporate more than one mRNA in the LNPs. Our CMV vaccine has, 6 different mRNA in the same drug product, and the EBV vaccine has also a large number. Do you have in vivo data in animals showing duration of mRNA expression? Yeah, so yes, we have. So yes, sorry. The question is, if we have really data showing in vivo expression at the site of injection. So yeah, we have performed experiments, you know, in mice mostly, and in non-human primates, and really the protein, it's in the genital lymph node is expressed, you know, up to 72, 96 hours. Really depends also on the antigen, but it's a transient expression. Do the nanoparticle fuse with the muscle cell after IM injection? Is there a specificity of, you know, or captured by immune cells? Yeah, so basically we don't really, what I can say is that we don't really transfect muscle cells, but the mRNA LNP transfect mostly, you know, immune cells, such, you know, like monocytes and macrophages and dendritic cells, but we're not the muscle cells. I'll pick some random just so that I'll try to cover. Yeah, the choice of the antigen at the beginning was critical. How many people were involved in to make the decision? Oh, sorry. Yeah, in only 2 days. Yeah, I think, you know, we were lucky, and this is important as this concept of pathogen preparedness. I mean, we had performed work on other coronaviruses and specifically on the MERS. So we knew what really construct we needed to express. And, you know, maybe there were discussions if this had to be, you know, at the beginning of the pandemic, it was not clear that this was going to be a pandemic. And to some extent, it was almost an exercise to show how quickly we could go. So we were discussing if this had to be a pan-coronavirus vaccine and maybe consensus sequence, but we, you know, quickly agreed to really settle on the sequence of this circulating SARS-CoV-2 vaccine. So, you know, in terms of decision on the sequence, it was relatively simple. We had to verify, of course, experimentally that we were getting the protein expressed in the right conformation and so on. But we basically, you know, initially down selected 7 or 8 candidates with the, you know, the wild type 2p mutant being the leading candidate at that point. So you tell me when you wanna go, but I can.
Oh yeah, is they're quite random. Some are quite similar, but yeah, I prefer to let
yeah, more and more variants are predicted to emerge. What is your capability to adapt, especially in terms of manufacturing? Yeah, so we can adapt very quickly. You know, for instance, for the new, you know, vaccines that we are testing in the clinic. You know, once we decided to really work on it, you know, it took less than 50 days to be in the clinic. I think it was 45 days. So in terms of manufacturing, we can adapt very quickly. Of course, you know, you have to still, you know, perform clinical studies, right? To at least look at immunogenicity, and the scale up, it's the other part that is going to take time. But I think the effort that we're doing at this time is really to understand, you know, what kind of immunoresponses we get with the variants and what is the best strategy, you know, moving forward. But we think we can address the variants, you know, very quickly as they emerge. In case, especially, there is a variant that is not, you know, neutralized by the current vaccines. Again, so far, I think the vaccines remain efficacious on the variants. Do you have people having received classical vaccine at the first shot and then variants at as a second shot? Yes, that's one of the study that is ongoing. So people that have received 2 doses of the Moderna COVID-19 vaccine, and now they're receiving a booster, so a 3rd dose that is one of the variant, or a mix again of the current vaccine and the variant. So we'll see what is the best approach. So
If you want, Andrea, you can stop answering the question because you have a lot of questions, and it's 4 o'clock here now.
Okay. So yes, I think I'm good. Yes.
Thank you very much because you have a lot of questions, of course. Because
I haven't looked how many.
Really
yes, it's interesting. There's a lot of interesting.
Yeah. Just maybe one general question: do you think this breakthrough with the mRNA vaccine is the end of other platforms, or other vaccine platforms, such as subunit vaccines or viral vector vaccines based on a viral vector? Or?
Yeah, it's an interesting question. I mean, I think in the different technology have different advantages. So I don't think it's necessarily, so the mRNA LNP approach will overtake all kinds of vaccines. But in many cases, I think it can, right? But for instance, adjuvants, some subunit adjuvanted vaccine, still have interesting features in terms of immunogenicity and protection, and I think they will still be are very useful for certain populations. I think also in the context of the pandemic response, the adenovirus platform was useful in the it's working also well, and provides protection also with one dose. So, but I do think that there will be a larger investment in the mRNA technology, and you can see that with the majority of large pharma getting in this space at this point.
Not only in vaccines, so also in immunotherapy or in inherited disease, for example? Or?
I think so. Right now is of course the focus for the scientific community, and the proof of concept, I guess, in humans is with vaccines. So I think that's where everyone is focusing. Personally, I think that in the future we will see also success in other areas such as oncology, rare disease, autoimmunity, and so the number of applications it's really huge, and is that clearly the technology will evolve and will become better, but I think that we will see more opportunities. Certainly Moderna is investing a lot on other therapeutic areas as well.
Okay, thank you again, Andrea. Maybe Winfried, you want to close the seminar?
Yeah, I just have maybe one final question. Do you see in all these studies, do you see immunogenicity against the LNPs?
No, I mean, we don't really, in the context, we don't really look for that, but we don't really see, in preclinical studies, we didn't see really responses against the LNP, yeah. And this is actually shown maybe even from the, boostability of the vaccine. For the CMV program, we have gone with the 3 doses. And for each dose, we see really a boost in the responses. It becomes more important of course, for the therapeutic application.
Well, thank you very much for this very interesting seminar.
Thank you again.
Hopefully your company boosts up production of your vaccine in Europe very soon, so that we can also profit a little bit more from that.
Yeah, I think that's hopefully I think, I don't want to, it's not advertisement. I mean, I think there was Moderna, the European community has signed for 300 million doses of the Moderna vaccine by Q4. I think that the doses that were, agreed are being delivered in time. There was, I think, a delay in the agreement, when Europe decided to invest on the Moderna vaccine, and so that's also led to some delays.
Unfortunately. Yeah. Okay, thank you very much, Andrea.
Thank you again. Thanks.
Thank you.